Welcome to San Diego State University. Please take your seats as we begin this morning's program. Please welcome San Diego State University's president, Dr. Elliot Hirschman.
Well, good morning. It's my great pleasure to welcome you to the first lecture in the San Diego celebration of the 2015 Kyoto Prize. The Kyoto Prize Symposium is supported by the Inamori Foundation, and I want to convey our gratitude to the representatives of the Inamori Foundation who join us today. Thank you very much for your support. The symposium celebrates the recipients of the Kyoto Prize, Japan's highest private award for lifetime achievement. The prize is presented annually in three categories: advanced technology, basic sciences, and arts and philosophy. With our colleagues at Point Loma Nazarene University, the University of San Diego, and UC San Diego, we're proud to bring the Kyoto laureates to our binational region. We're especially grateful to Dr. Inamori and the Inamori Foundation for providing this extraordinary opportunity to our region. And now we're going to see a short video illustrating the nature and significance of the Kyoto Prize.
The annual Kyoto Prize ceremonies take place each November in Kyoto, Japan. The Kyoto Prize is Japan's highest private award for global achievement. It is given each year by the Inamori Foundation, which was founded in 1984 with the initial private funds of Dr. Kazuo Inamori, founder and chairman emeritus of Kyocera Corporation. Human beings have no higher calling than to strive for the greater good of humanity and society. The Foundation awards 3 prizes annually in the following fields: advanced technology, including electronics, bio and medical technology, material science and engineering, and information science; basic sciences, including biological, mathematical, earth and planetary sciences, astronomy and astrophysics, and life sciences; and arts and philosophy, music, arts, painting, sculpture, craft, architecture, design, theater, cinema, thought, and ethics. The Kyoto Prize Symposium is held in San Diego, California, to share the ideas of prize laureates across the globe. The philosophy of the Inamori Foundation aims for a future with the proper balance between science, technology, and spiritual maturity, with the goal to contribute to the peace and prosperity of humanity. This is the core mission of the Kyoto Prize.
Before we begin, I'd like to recognize and welcome a number of distinguished guests. First, our 2015 Kyoto Prize laureate in advanced technology, Dr. Toyoki Kunitake. The laureate in basic sciences, Dr. Michelle Mayor, and the laureate in arts and philosophy, Mr. John Neumeier, are unable to join us today. They're preparing for their talks later. Next, we're very fortunate to have 2 distinguished leaders of the Inamori Foundation with us. Senior Managing Director of the Inamori Foundation, Mr. Toyomi Inamori. And the Executive Director of the Foundation, Mr. Shoichi Himono. From Kyocera Corporation, Mr. Kaz Umemura. very fortunate to have leadership for the symposium in our own community. The founder of the Kyoto Symposium organization, Mr. Malin Burnham. The director of the Kyoto Symposium organization, Mr. Rod Lanthorn. And the executive director of the Kyoto Symposium organization, Mr. Dick Davis. Also very fortunate to have university leadership join us today, Provost and Senior Vice President, Dr. Chikuka S. Nwemeka. And Vice President for Business and Financial Affairs, Mr. Tom McCarran. And finally, at San Diego State, we always like to bring things back to the students. I'd like to recognize the extraordinary SDSU graduate students who received the Inamori Fellowship this year. Could you please stand so we could recognize you? And now we will see a special video highlighting Dr. Kunitake's achievements.
Toyoki Kunitake was born in 1936 in the Fukuoka Prefecture of Japan. He is the eldest of 3 brothers. His mother loved books and instilled in her children the joy of reading. Toyoki's father ran a grocery store and looked forward to his children attending universities. Toyoki went on to major in applied chemistry in the engineering department of Kyushu University. As a student, he developed a passion for research and decided to pursue a career in research and development. After completing his master's program, he traveled to the U.S., where he earned a doctoral degree at the University of Pennsylvania and became a doctoral fellow at Caltech. He later rejoined his alma mater as an assistant professor in the engineering department of Kyushu University. Biology shows that the walls of a living cell consist of bilayer membranes. These membranes have hydrophilic and hydrophobic parts, as their elongated molecules face each other and line up in highly organized layers. Individual molecules come together spontaneously, organizing into a regular structure through a process called self-assembly. For decades, researchers believed this phenomenon occurred only in living cell membranes. Then, in 1977, Dr. Kunitake produced a breakthrough. Through a process of synthesizing lipid molecules, he discovered that this self-assembly phenomenon could create synthetic bilayer membranes similar to the membranes of living cells. Dr. Kunitake also worked on a methodology to immobilize bilayer membranes for specific applications. He helped establish a new field of chemistry based on molecular self-assembly and immobilization technologies. This has opened a new frontier in polymer materials science and supramolecular chemistry, leading to new technologies and materials that are now widely applied and commercialized. Dr. Kunitake currently serves as president of the Kitakyushu Foundation for the Advancement of Industry, Science, and Technology, where he remains extremely busy supporting national research projects and cultivating corporate partnerships. To young people, he gives the following advice. There is no limit to knowledge or technology. Please become humble optimists to create a better world.
Please welcome San Diego State University's distinguished professor and chair of the Department of Chemistry and Biochemistry, Dr. William Tong.
Good morning. In 1977, way before most of the students here were even born, even some assistant professors, Dr. Kunitake published this electron micrograph picture you saw of the synthetic bilayer membrane in the Journal of American Chemical Society. For those of you who don't know, that is the most prestigious, one of the most prestigious journals in chemistry. And this was, this major breakthrough, this discovery he made was only 14 years after he finished his postdoctoral research at Caltech as a relatively young scientist. Now, our students in this room are really lucky today because it's not every day you get to meet a giant in our field, but also somebody who actually created and changed a major field of chemistry. Now, I remember a generation ago, another group of SDSU students met Dr. Glenn Seaborg, Nobel laureate Glenn Seaborg, and it was actually in this spot, even though it was a different building. So Dr. Seaborg achieved immortality because his name is on the periodic table. Look it up, Seaborgian. Okay. Now, Dr. Kunitake has achieved similar immortality because, I'll tell you later, all these chemistry students in the future will be studying his projects, okay, in your future textbooks. Supramolecular chemistry. Okay, remember that. This is an area of chemistry where it's way beyond molecules and traditional covalent bonds that you have learned in high school, okay? I won't get into the detail, but there are some interesting YouTube videos some of our students might want to download and look at, okay? So one of them was posted by the Scripps Research Institute, and where they have a little plastic bottle and full of randomly shaped Lego-like pieces where you shake it and simulate some energy, temperature, interaction, and eventually they all kind of form like a perfectly round ball, okay? Now, this is an oversimplified version of what is going on with self-assembly, but you get the picture, right? Now, of course, Dr. Kunitake has made molecular assemblies that are really fancy with custom sizes and shapes and thicknesses and actually mimicking a living organism that can produce really complex molecules like proteins, right? Now, you've heard of nylon and Teflon and Kevlar and all these materials, right? Now, they're so 20th century, right? Now, look out for these new materials coming out in the future, okay? A lot better, with more exciting applications. And for example, molecular magnets and molecular architectures and nanofilms that are only tens of nanometer thick, okay? And yet they're very strong and flexible. Now, think of a soap bubble, right? And it's a film, but it's not strong, right? So what he has created is really strong and really flexible and really small. And if you stack up about 10,000 of these, they will add up to about the size of the paper in front of you, okay? So what are the potential applications with global impacts? Well, how about making fresh water out of seawater inexpensively, right? We all need that here in Southern California. And using his membranes, we'll be able to do that with less energy. And how about fuel cells? Okay, we can make them with a lot better efficiency. How about molecular-oriented film electrodes that can be used in vitro diagnostics? Okay, and all kinds of new materials that has applications in new materials for electronics, drug discovery, polymer materials, and everything. Now, this is the age of bioatomics, right? And he is building bridges, right? Building bridges by synthesizing stuff that go across boundaries, right? Across a wide range of sizes and scale and materials. And he's actually indeed building bridges across disciplines, from physics to chemistry, to biology, to engineering. Now, 55 years ago, when the laser was invented, nobody could have predicted all the applications we have today, right? From telecommunications to medicine to diagnostic tools to even entertainment. Now, I've been using lasers for 40 of those 55 years, and we're still discovering new ways to use lasers today. Now, similarly, Dr. Kunitake's discoveries will be used in future applications that we haven't even thought about. Okay, we couldn't even predict today. So finally, a quick message to all the chemistry students here related to his research. Think of this seminar as your first lecture, okay, in these new topics. The American Chemical Society, as you know, is meeting downtown at the convention center this week. And the department chairs for chemistry, we all met. We all had a meeting a couple of days ago. And the American Chemical Society is deciding, requiring that all the chemistry majors, if you want an American Chemical Society certified bachelor's degree in chemistry, you have to study the following topics. Let me read. Synthetic polymers, biological macromolecules, supramolecular aggregates, and nanoscale materials. Now, sounds like the American Chemical Society just got the message from Dr. Kunitake. And all future students, we have to offer required courses, not just elective anymore, okay? So just like quantum mechanics, Einstein's equations, and all the foundation topics, you're gonna have to learn his research. This is what I call immortality, and this is a long-lasting legacy. So, and now it's a great honor for me to introduce our 2015 Kyoto Prize laureate in advanced technology, Dr. Kunitake.
Thank you very much. It is a great pleasure to visit this campus. This is a beautiful city and a beautiful campus and marvelous lecture room here. So this is, as scientists, we usually spend humble lives. Our lecture room is not that good, but this is marvelous. Unfortunately, the good thing about my research has been given already to you by previous speakers. So I was wondering, by sitting in front, that what I should talk about. So I would like to show you some of how I became enchanted by, by science, how I got interested in science. And this is fundamentals of science are simple, straightforward. Once it is known, you see, it's easy to understand the fundamental aspect. So I like to concentrate on the fundamental aspect of research and of my work. First of all, I'd like to introduce you where I come from. I come from Fukuoka, Japan. It's the southern part of Japan. And we have about the same climate as San Diego, being in the south of Japan. However, Japanese climate is much more humid. We are going to have a humid season, rainy season soon. But here, in California, is very different. Oh yes. You see, and it take 16, 17 hours between Fukuoka and San Diego. This is very long trip, and so I would like to step by at Hawaii on my way back. That is very nice, enjoyable. Should be. Well, I work at the Kitakyushu at the moment for helping, promoting the collaboration of industry and academia. This is very important because as the industry changes with age, and this city started heavy chemical industry about more than 100 years ago, starting from coal, making steels from coal and iron ore. And because of the long tradition, many of the industries going down and had new problem because of the new, by the progress of information technology. And I'm, as a president of this foundation on the Kitakyushu, I'm sorry. We've been working on how to find new technologies, new products for the local industry. This is the kind of work I'm doing I'm spending most of the time. Well, now comes this picture. I was born in 1936. It's amazing. So I turned just 80 this year. Many people think that I'm too young for that age. So I'm say, sorry, I can't make it myself. And maybe the reason for that is one thing is good health given by my parents. The second, the mental situation. I'm quite optimistic. And this was good for research. Research, it can be done only by optimistic mind. Otherwise, you never try to do new things. Well, I'm in the center. And this is after only 1 year after the Second World War. And so Japan was quite in a very humble situation. And we didn't have enough food or enough clothing at the time. But this is summer, so we could run about in the neighborhood by by pants, and nobody. This is quite usual, and this is my mother. I think I inherited the interest, curiosity from my mother, although she didn't have higher education, but she was interested in reading and things like that. And I had two brothers, yeah. Then this is a high school picture of myself. And well, we used to have cap. That was a part of the uniform. And this, I'm sorry. This is not pointer. I thought it was a pointer, this antenna. I'm in the front. I'm a high school student. During the high school period, I was not sure at all what I should be in the future. I liked reading books, literary books, and I was thinking I might be studying economics or Japanese literature or medicine or anything. Just I was interested in just about anything, and I didn't really think that I would be a research chemist at the time. But at the final year of the high school, we had to decide what kind of discipline we choose by entering college, and I had to choose. My father at that time suggested me to become take a pharmaceutical science major, and that is because pharmacy shops nearby my house were doing good jobs. So my father must have thought it is a stable, good job to live on. But I was not interested. I myself was not interested in that. That by selling pharmaceuticals to the local people was not exciting to me at that time. And so I persuaded him that pharmacy is based on chemistry. So I can go to the chemistry major. Then I may become a pharmaceutical people later on. So that's why I could enter the college, university, majoring engineering and chemistry. Engineering was interesting because this is around 60s, and new industry was just coming on. It's the middle of the 20th century, and in U.S. particular, the petrochemical industry really exploded and became a big industry, industry worldwide. And so I wanted to be one of part of it, and studied at the senior year. I chose polymer chemistry, doing senior research as working on the polymer chemistry. And then I finished that B engineering, Bachelor of Engineering in 1955, and subsequently Master of Engineering 1960, and again working on polymers. New polymers was popular. You mentioned many plastic materials are abundant, commonplace anymore now, but at that time, new polymers, new materials are coming out from the petrochemical industry. So this group started working on synthesis, making new polymers or developing new polymers. When I finished, I was in the graduate school. I wanted to become an industrial chemist because that was a good way to live. And my father had died in my freshman year, so I had to support my family. So I wanted to earn the money as soon as possible. But fortunately or unfortunately, my professor, my mentor professor at the time, suggested me to go to study abroad in the U.S. to receive better new education, because our education at the engineering school in Kyushu or in other Japanese school was more or less process type of process, how to make materials, how textile, dye stuffs, kind of thing. And so we professors thought that we need more fundamental, broader knowledge to develop new things. So he suggested that, but he didn't mention which school or what kind of thing, university, what kind of school to go. So I wrote application letters to several professors in the U.S. One of the professors was Charlie Price at the University of Pennsylvania. He was a very famous polymer chemist at the time. And he told, yes, you can come to join a research group. I can provide you research assistantship. So that was very nice. And for travel expenses, I obtained a Fulbright scholarship Well, Fulbright scholarship is not well known in this country, but that was very well known in Japan, in the post-war Japan especially, because Senator Fulbright at that time, he was a diplomatic expert in the Senate, and the money. The U.S. provided foodstuffs to Japan, the post-war Japan, and the money for that was he proposed can be returned as fellowship, scholarship. So this Fulbright scholarship, I could obtain travel expenses by Fulbright scholarship and living expenses in Philadelphia by assistantship. So I was very lucky to be able to study in Pennsylvania. The upper picture is a part of Philadelphia, or a building of the University of Pennsylvania. And I took again synthesis or preparation of new polymers as a research topic. And my experience in Pennsylvania was very valuable because I had to receive, I was given a very different kind of education, chemistry education mainly, and that is more or less discussion-based, or discussion and presentation are more important, or even now it is the same. In this country and back in Japan, the same thing happens now. After 2 years, more than 2 years, almost 3 years, I could finish my PhD process at the University of Pennsylvania. And a good thing about it is Professor Price had a talent in many areas. He was a good politician. He even tried to become a member of the Congress, he did succeed in it. But he was a good professor and had a good management capability. But because of that, his talent, he didn't come to the lab often. So we could do our research more independently. That's a good part of life at that time. And after 3 years I wanted to do something different. And I was interested in chemistry and biology, just interest at the time. So since I had been working on chemistry, chemical synthesis of polymers, I wanted to change my subject a bit and found that there's a postdoctoral position at Caltech on enzyme kinetics, enzyme chemistry, how to study how enzyme works. Down here, the picture, this is a part of a Caltech, I did the postdoctoral fellow. I was there between 96 to 63, working on kinetic study of the enzyme catalysis. And at that time, enzyme was a protein, that is a part of protein, and we already knew that enzymes are good catalysts, which we cannot explain well by simple chemistry. So many people are interested in why enzyme is good, so much good, so much better than the counterpart, artificial catalysis. And this experience was very important. And so Caltech postdoctoral process gave me some knowledge in biochemistry. So I could combine in the following years my knowledge of chemistry, synthetic chemistry, and enzyme action kind of things. The second thing, that is this Caltech experience was good or that. I lived in Pasadena, and I met my wife in Los Angeles, and we got to know him, and came down to San Diego for a date. So this is my second or third visit to San Diego. And it is unbelievable that that relationship continues for more than 50 years now. So by chance, accident, or incident is very important to be in science or in life. Now, I go back to the Pennsylvania age. Negishi, Professor Negishi, he is now a professor at Purdue University, chemistry department, and he obtained Nobel Prize in 2010. And we were together, a fellow graduate student at Pennsylvania, and we worked in the same room for making new synthesis. And we didn't have close contact since that time, since the graduate period. But he, after obtaining his Nobel Prize, he came to Japan, he visited Japan, and our government started this area must be important, his area must be important, and promote by funding, establishing a new fund. And that is what we call Act C. Act C means this is a chemical transformation project, chemistry really, chemical catalysis, and CO2 including reduction or transformation of CO2. That's related to environmental problem. And I was made a general manager of this project. It's a huge, and Professor Negishi helped me, advisor. And it includes some more than 50 research groups all over Japan. So it's a huge project. But it is interesting that after, after 50 years, we could make a close contact again. So we talk each other. What life is strange, very strange. You can never lose what you experience. What you experienced in the past may come back again after so many years. Well, I went back to Kyushu University after finishing my postdoc period, and I started on research on, again, in the polymer areas. But synthetic polymer was research activity was going on worldwide. But still, the heat we felt during around the 50s was going down because many interesting polymers has been synthesized, and making new polymers was not easy. Appears to take a lot more effort. And the interest of the people turned slowly to the functioning of the synthetic polymers or new polymers. It's a chemical function, like biological enzyme work. So, as I mentioned that, I was interested in chemistry and some biology area. So I wanted to combine two areas, or to work on the interfacial area of research. And the biomimetics was just coming into the chemical research at that time. This is around 1960s. the enzymes, how enzyme works in the body, it's getting clear now at the time, only at the time. But protein structure, complex protein structure was not yet known. The first protein structure, that is myoglobin, was determined in 1960 by X-ray crystallography, and that was worth of the Nobel Prize. The first protein, the structure was complex, very complex protein molecule became clear for the first time. Well, anyway, biomimetics is defined as to imitate the shapes and functions of living things in order to solve complex human problems. Well, biology or biochemistry try to help you make in health problems and things like that, or food problems. And the biomimetics want to do the similar thing by artificial means, by artificial materials. And I just give you a recent example of the biomimetics. You see, this is a morpho butterfly with very vivid colors. And this color these colors don't come by pigment. It comes from reflection or interaction of light. And why this color comes from is based on the particular structure down below with regular layer structure is a cause of this color. And this is what are recent findings, and we call it structural coloration, not pigment structure. The structure itself provides colors. And this is another example. Jewel beetles, it is called in English. It's colored, very well, nice colored insect. And again, these beetles have a layered structure on the surface. And the layered structure, again, in the same way, because of the structural coloring effect, this color appears. And if you see the surface by electron micrography or high resolution optical micrography, you see unique layer structure on the surface, as is shown here. Well, I just give you a typical example of light reflection and interference of light. Of course, a rainbow is, you see 7 or 8 more colors because of the reflection of the light, depending on its wavelength. And blue light scatters much better than the red light kind of thing. It's common knowledge. And the interference is a balloon, water balloon, soap, solution balloon, and where the very thin layer of water is formed as a shell, or the balloon shell, and because of the light, wavelength of light, it becomes closer to the thickness of the shell, then color interference happens. It's called the Newton ring, but it's colored. It changes the color, like jewel beetles. And this, I'm sorry, I have examples. I wanted to wear this tie, but it is not very clear. This tie, if you look closely, it's colored. Well, they can make it more well colored, but actually, this is called the morphotext. This is made of fibers, layered structure, with layered structure. And because of the reflection or interference of light, they can create color without using a pigment. You cannot say, but okay. Just believe me. Well, you can buy, of course, and test it yourself. Well, the one of the biggest problem of the biomedical chemistry is depends. It depends on the size of size level of size, how big it is. At the molecular level, how to mimic the protein action, or how to mimic the biological cell function of biological cell is typical target in the biomedical chemistry at the molecular level. But higher level, you see many examples of biomimetic efforts, and this is the one I just showed you. That's color. Structural color is an example of in the range of wavelengths of light. At higher level, you see many examples like gecko attaching to the ceiling bracket, and that is because of this interaction of very small space to the surface. What we call van der Waals interaction appears, and it can hold the weight of those gecko and things like that. There are many examples in nature of that kind. Well, coming back to the molecular level, you see this is a rough picture of a biological cell. Biological cell is made of the surface surface is biomembrane, and biomembrane is composed of lipids and protein combined nice and organized, making nice layers, films, and inside this cube there are many structures called organelle, which does chemistry or physics in the biological cell, like mitochondria that helps us to produce energy from materials. ATP for example, ATP is a source of energy for our muscles work, and the nucleus is a center for information, and they have gene replication and translation is done by its nucleus, DNA provided from the nucleus, or protein synthesis is done by a ribosome, and the lysosome is used for dissolving or degrading the useless component within the cell. So biological cell can do a lot of whole thing just by one cell, small cell. It's a micron size. And this is unique. So the target of biomimetic chemistry, as I said, is this cell is one of the best targets in the biomimetic chemistry. But it's not easy to reproduce the whole functions, whole structures by simple artificial materials. This one is you may know that this is a molecular structure of a globular protein which is round-shaped. And this kind of protein is made of long peptide chain, which is folded into a particular shape. And this particular shape can be never produced by synthetic polymer, because synthetic polymer may hold into a small cube, small particle, but it's random process, and you can not say the exactly formed structure based on that synthetic counterpart. So what chemistry? Well, I mentioned that I worked on enzyme catalysis, and on chymotrypsin, and so what, as biomedical chemistry, what I did first at the Kyushu University is enzyme model at Kyushu University is that we synthesize simple polymers which contain one functional unit, the other second folding unit. This copolymer A here is, and we published the result to American Journal of American Chemical Society, and proposing that kind of random polymer chain, even the random polymer chain, can hold partially to fix the substrate, the reacting species, and then perform catalysis. This folding model is a very rude model of enzyme action, but still, it works. Its kinetics, its rate process is similar to that of enzyme. So we had nice beginning start in the biomedical chemistry area. But as I said, that unfortunately we could not reproduce the more precise structure of enzyme, because most of the enzyme action comes from that very specific structure. We could not reproduce it. We could mimic in a very rough way only. So this work took about 10 years for us. There was a worldwide competition in this area. It was okay. But in 10 years, this area sort of stopped growing because of the limit of the artificial mimicking technique. So again, we go back to the biological cell itself. We could, in 1977, Singer and Nicholson in the US published a paper on biomembrane. It's a model, the model is called fluid mosaic model. It's fluid mosaic model is this, this. Their model was two lipid molecules bilayer, so two-layer, bilayer, forms flat aggregation, assembly to form membrane structure, basic membrane structure. And the protein is immersed in that bilayer structure and moves around for functioning. This is fluid and the mosaic. Protein molecule is in the mosaic on the surface of membrane. This proposal was 72, and this model is thought valid, valid, and people use this model even now. This is a basic model of the biomembrane structure. So, oh, sorry. Well, so we thought we wanted to make the artificial component better organized, and we didn't succeed in making the long chain to fold organized to be better organization, like protein. It was not possible. We didn't succeed, and it is not possible even in the present time. So this is a big research target, but we cannot do it. The biomembrane is characterized by molecularly thin, two molecules layers. So it is the thinnest flat membrane available, including artificial systems. And various components immerse it and cooperate to do various different functions, versatile function. And even more, it is self-supporting. That means both sides of the membrane is water, an aqueous phase, and the membrane is stable by itself in water. So it has self-supporting, freestanding nature. So these various unique feature of biomembrane is a good target for biomimetic chemistry. Well, since we got interested in this biomembrane mimics in 75 or 6, a few years later, after the real true biomembrane model was proposed. And if we think from the synthetic chemical side, then the nature, characteristics of biomembrane is based on two things, two factors. One is stable supramolecular interface. It has to be aggregated, make stable structure. Supramolecular. Oh, you mentioned about supramolecular. And the second, within the molecule, molecules are organized, ordered. So ordering is important. So we have to we have to make include in the process ordering and supramolecular interface. Well, these are the top typical phospholipid included in our body, in the membranes, biological cells in our body, phospholipids. And phospholipids molecules have a structure as shown this. There are various kinds of phospholipids, and there are small, slight changes in structure. And these lipids are mixed in the biomembrane to form, in the bilayer, to form nice stable membranes. And this lecithin is the best known lipids, maybe. And we applied our new thinking to this lecithin molecule, structure of lecithin molecules, and decided to provide synthetic counterpart to the of the native lipid molecule. The lipid, this is in Japanese. It says phospholipids. Yeah, they are up. I'm sorry, I forgot to change this to English. But anyway, you would understand this. The upper structure is a native one, lecithin, structure of lecithin molecule. And we decided to simplify this structure from the synthetic, based on the synthetic view. And down below is a synthetic counterpart we use, and that is simplified. The natural lipid have synthetic hydrophilic, water-friendly units on the surface and oil-friendly hydrophobic units as a tail. The two portions are combined. And so we use simpler hydrophilic head group and a similar tail group and combined as synthetic model. And when we dispersed that, we put this white powder, the synthetic counterpart, white powder. When this is put in water and sonicate or heat somewhat, the solution becomes clear. So, and when this is observed by electron micrography, we have to see the molecular details. So electron micrography was the answer to see that morphology or structure. And on the which way? On the left, yeah. On the left, you see one example of electron micrography. And this is a picture. Electron micrography gives you a sort of dried squid picture as against live squid moving around. But still, you can guess what will be live structure based on the dried structure. And you can see spherical object, and inside black part is the originally water phase, and outside is again water phase. And you see membrane structure and thickness of this in this electron micrography is similar to the 1 bilayer or 2 bilayer in range. So we are very happy to see this picture. That simple molecule. You see, this compound, synthetic compound is, this kind of compound is used for conditioning your hair. Some of these has been commercialized by Eastman Kodak. And when I showed this result to the people, research lab, at the research lab at Eastman Kodak, they are very much upset because they had enough materials to work on, but he don't think that this is possible. Well, so idea is more important than material. Anyway, once you know that this spontaneous formation of the structure, supramolecular structure, membrane structure is possible, then you can extend that concept to other systems, related systems. I just mentioned water bilayer membrane formation in water that is discovered. We published it in 1977, 4 or 5 years later after the biological proposal on the biological model. And since this basic point of that structure formation is stability, supramolecular stability at the cell interface and the molecular organization within the membrane. So if two conditions are satisfied, then we should be able to make the similar structure in other systems. The second one here is in organic media. We made a new compound that is composed of polyfluorocarbon. Fluorocarbon is not miscible with gasoline, hydrocarbons. And the flexible tail, head group, flexible one-chain head is miscible with organic solvent like gasoline. And this kind of compound forms bilayer structure even in organic media. And because head group unit, flexible unit is miscible with solvent, and fluorocarbon components are not miscible. They aggregate in organic solvent, forming membranes. Or in the third case, we can separate this membrane structure into two parts, hydrophilic part and hydrophobic part, and connect them by hydrogen bonding, proper hydrogen bonding, like nucleic acid bases. And then this again forms nice bilayer structure in water. In this case, it's in water, but each component made of two separate different components, each unit, membrane molecular units. So now we see we started from biomimetic chemistry, but after finding these results, we thought this is based more on the physical or chemical principles of molecular interaction. So it's not limited to the biological world. We can use this for synthetic world. Well, this self-organization in nature is quite interesting. So our research interest is turned more and more to the self-organization of molecules. And I show you here the examples of self-organization in nature. The tornado is helical winding of the air or wind. is a typical example of self-organization of the flow of airflow. Or the central one, oh yeah, down below, a galaxy formed is considered to be a self-organized structure, a system. And the central one is, we call Belousov-Zhabotinsky reaction, based on the Russian scientists who discovered this phenomena. And they found two components react with each other, forming different colors. But because of the diffusion rate and the relationship of diffusion and the reaction rate, they form this ring, multi-ring structure on water surface. And also, of course, leaf vein is one of the self-organized systems in nature, because its vein is spread out to all over the leaf so that they can provide water and get a product from the leaf, individual part of the leaf. The last one is skin, human skin. If you look at yourself very closely, you will find this kind of self-organized structure on surface. Well, this has meaning itself, biological meaning. So, well, time is up now. So I would like to, my research interest started from biomedical chemistry in the 60s. And we wanted first to use that concept to make enzyme-like molecule. But we have only partially succeeded in that effort. So we then turned better organized structure, that is membrane, bilayer membrane of the biological cell. And we could show that process, membrane formation, is quite general. It's not limited to the biological world, but more on the chemical or physical systems. And biology, nature, use very carefully only the available material for making their own bilayer membrane. But synthetic chemists can make new materials, and they can, so that they can give you new type of bilayer membranes. And we have made hundreds of these compounds. That effort lasted almost 15 years, and the student just kept working. Well, I also did, but mostly students. But finally, so we could sort of general conclusion for that topic. And now we are coming to the next stage. What is possible by making very thin membranes, like molecular thickness, thick membranes? Because that is important, as you could introduced. And if you provide a very stable, large-sized molecular membrane, then selectivity is good, then transfer of the materials becomes much more efficient because of it is thin, much less resistance against membrane. So many chemical processes can be more energy efficient by taking advantage of these membranes. We've been working on gas permeation across the very thin a few tens of nanometer thick membrane, and still it's not good enough. Biological membrane either have a thickness of few nanometer, so we can obtain a large stable membrane with about 10 times thicker ones by artificially. So we have to go down further and make them stable enough, good enough for using industrial purpose. Well, finally, I would like to talk to many of the students. What is research so exciting? I've been excited all over the year by research, sometimes forgetting about family. I'm sorry. Well, I see several aspects of excitement in research. Of course, the new technology, new science is useful for solving world problems, various problems we face, environmental problems, energy problems, other problems as well. And that's a very serious part of the public part of the excitement. But also there are personal excitement. That is, well, it is provided, for me, it provided personal satisfaction like reading mystery novels. You grow, you don't find the answer from the beginning. You sort of muddle through the mysteries of science. That process is interesting, like reading mystery books. And also, if you get a very nice result, a new concept, then it's like conquering high mountain, climbing up Everest first time. That's a great achievement. So this two things is a source of personal excitement, and this excitement could have made me work quite hard over the past almost 50 years at the moment, until now. Well, so I hope that younger generation will understand you can have personal satisfaction as well as public satisfaction. And personal satisfaction is important driving force for working in this sometimes complex science. Thank you very much for your attention.